Okay, welcome everyone. Thank you for coming by. I can appreciate it. It's quite early, at least for me. I don't think I've ever been awake after an open bar this early. We're going to have a list of very exciting talks today. We're going to start off with a presentation by Sheena Jocelyn. Unfortunately, she couldn't make it in person, so we're going to watch a recording. She will talk about engram, the history of the engram, and specifically talk about her latest research in rodents. Then afterwards, we're going to have Joshua Jacobs, who came all the way from Colombia, who's going to talk about spatial navigation and phase precession. Then we're going to have Florian Moorman, who will talk about concept neurons as the building blocks of semantic memory. Say again?
Semantic building blocks of episodic memory.
Episodic even? Okay. Sorry, episodic memories. And he's going to also present some pharmacological studies. And I will round it up with episodic specific neurons that we found in the hippocampus that code for specific episodes. So glad you're all here. Let's get going.
Hi everybody, I'm Sheena Jocelyn and I'm delighted to be telling you today about our lab's work entitled Hippocampal Neuronal Allocation to an Engram Supporting a Memory in Mice. So my lab is really interested in understanding how the brain encodes, stores, and uses information. And because these studies can't be done in a human brain, we're going to be using the next best thing, maybe, a mouse brain. So we're going to be looking at memories and how they're encoded, basically a memory trace or an engram. So our first question was, how are experiences, or how is a memory represented in relevant brain regions at the circuit or neuronal ensemble level? And it could range from a very, very dense code where every single neuron is engaged all the way to a very, very sparse code, very much like a grandmother cell. And many studies suggest that it is a sparse code. So we said, okay, well, how are neurons within a brain region allocated or recruited to this sparse engram? And to examine this, we're going to be looking at a fear or threat conditioning. There's two varieties: auditory fear or threat conditioning, where we pair a neutral tone with an aversive stimulus, such as a mild foot shock. Then we can test the mouse anytime later, and we just replay the tone and say, do you remember the last time you heard this tone, you received this aversive foot shock? And they show us good memory for this by freezing or showing this defensive response to this tone. So in a series of experiments conducted over many years, we looked at how this auditory fear memory is represented in the lateral amygdala. So in a series of experiments, we have a bunch of neurons in the lateral amygdala, and we used viral vectors, each expressing different constructs over the years, but the common denominator is that they all increase the excitability of these randomly infected neurons. So we microinject these viral vectors. These neurons that are infected are going to have increased levels of excitability relative to their neighbors. And lo and behold, we found these neurons with increased excitability right before the time of training ended up becoming what we call an engram neuron, so a neuron that's really important in storing this memory. So these are very sort of correlational data, and they suggest that neurons are allocated to a sparsely encoded engram based on excitability at the time of training. But are these neurons really necessary? Are they critically involved? So to test this, we're going to use a very cool optogenetic construct, which allows us to increase and decrease the excitability of the same population of infected neurons using these two different optogenetic constructs. So if we shine a blue light, we're going to hit channel rhodopsin, an excitatory construct. We're going to increase the activity of these neurons. And if we shine a red light, we're going to hit halorhodopsin and basically silence these neurons. So this is what we did in the lateral amygdala to show that these neurons are critically important for later retrieval of this fear memory. So we're going to infuse a viral vector exposing both channel rhodopsin and halorhodopsin. It's going to infect a sparse random population of cells in the lateral amygdala. We're going to shine the blue light. We're going to allocate these cells to the engram. How do we know these cells are allocated? Well, we're going to test the mice under two conditions. Once normally without a red light, just say, what does the memory look like? And then when we specifically silence just these infected cells. And what does this show us? Well, under normal conditions without the red light, these mice show us a very high percentage of time freezing to the tone, shows us good memory, high freezing, good memory. Then when we silence just the small population of neurons we've tried to allocate to the engram supporting this memory, we see a memory deficit. And we do not see this when we do not allocate these neurons prior to training. So if we just silence a small population of neurons, nothing happens to the memory. So in this background, we say, okay, is this effect generalizable to other brain regions, specifically the dorsal CA1 of the hippocampus? And here we've manipulated the excitability of these neurons. Does this happen endogenously? So first, we're going to rerun the exact same experiment, only now we're going to look at a contextual memory. We're going to have two groups of mice, and one group, again, we're going to allocate those neurons. This group, non-allocated, we're going to test mice under two conditions, normal conditions. And when we specifically inhibit just these infected neurons, we get exactly the same profile, suggesting that this activity or excitability-dependent allocation happens in the CA1 of the dorsal hippocampus when we experimentally manipulate these neurons. Then we said, well, what's the profile? What does the activity profile look like when an animal is retrieving this memory? And for this, we're going to use GCAMP, a genetically encoded calcium indicator, just as a proxy of neuronal activity in these mice. And because we're going to be taking up the blue and the green channel, we're going to use a red light sensitive optogenetic exciter to allocate neurons. So again, we have two groups of mice. In one group of mice, we're going to allocate those neurons by shining the red light. We're going to try and allocate these neurons into the engram. The other group of mice, they're just going to be random. We're going to train mice in contextual fear conditioning, and we're going to test them. But during the test, we're going to read out the activity of all the cells that we've infected. Here, the cells allocated, we think are important in the memory trace. In here, the cells not allocated, probably not important in the memory trace. Memory is equivalent in both groups. And here's what we see. So when a mouse enters a freezing bout during the test session, each time it's one of these dotted lines, we see this increase in activity of these allocated neurons. And we do not see this increase in activity if we do not allocate these neurons. When we align all the freezing bouts to time 0, we really see this. This is at the population level. We're really interested in this at the level of individual neurons. If we find individual engram neurons, we're going to trace their excitability back to before the animals have been trained and say, hey, are these neurons that are more endogenously excitable, are they the ones that are recruited or allocated into the engram? So for this, we're going to use a mini endoscope made by someone in my lab, name of Chen, so we call them Chentoscopes, and it's open source, anyone can build them. So here's what we do. These are mice expressing, again, GCAMP. We're going to train them in contextual fear conditioning and test them. And this is going to allow us then to identify engram neurons and trace their activity back to before the mice were fear conditioned. So 5 minutes immediately before in the home cage, 3 hours, and also 24 hours before in the home cage. And we're also going to look at this in a different context. So based on their activity during the test, we're going to identify neurons based on their activity, classify them as either engram or non-engram neurons. And with this, we have about 7% of the neurons we classified as engram neurons. And first, we're going to just do a sanity check. So during our endogenous experiment, this is the profile of all the engram versus non-engram neurons. And this looks really similar to what we saw in our experimentally allocated group. And we don't see this when we test them in a different context, the novel context. So this really suggests to us that we've identified engram neurons, or neurons critically important in the memory, in our endogenous case. So what do they look like before a mouse has been trained? Well, 24 hours before in the home cage, we see no difference in the excitability between these two populations of neurons. During this 5-minute period, when a mouse has been placed in this novel environment, we see that engram neurons seem to be more excitable. And they're also more excitable immediately in 3 hours before. They are not more excitable in a different context test. So this suggests to us that these neurons are allocated to a memory trace or an engram based on excitability in the minutes to hours before training. Then we did a bunch more analysis. This is partly correct, but also pre-configured functional connectivity also influences allocation. So the brain is not a bag of neurons, that it comes to us with somehow different neurons being functionally connected. the ones that are more excitable, the sub-ensembles that are more excitable at the time of training, their connectivity, functional connectivity seems to be enhanced by training, and it is these neurons that become allocated to the engram. So just as a sum up, we found that excitability-dependent allocation does occur, and it occurs also in the context of pre-existing functional connectivity. So the brain is not a bag of marbles. It does come to us based with some pre-existing functional connectivity. And the most excitable sub-ensembles, these functionally connected bunches of neurons, seem to be the ones that are allocated to an engram. This activity pattern seems to be stamped in at training and recapitulated later at memory retrieval. That sub-ensembles may be formed based on previous experience, or maybe because of some hardwiring that we haven't yet investigated. And this is consistent with some notions of pre-play, or that there is some sort of pre-configured sub-ensembles with higher functional connectivity, or some structure upon which learning occurs. And with that, I just want to thank my funders, my collaborators. This is my husband, Paul Franklin, my lab. This is us on our pandemic selfie day. And to let you know that we are always looking for students and postdocs. And with that, I want to thank you for your time and attention. I'm sorry I'm not with you in Glasgow, but the next best thing. Thank you so much.
I think that was a very cool presentation. It's a shame that we can't do these things in humans, because I find the idea that excitability increases the probability of allocation to an engram quite elegant. Really cool stuff. So next up, we're going to have Joshua Jacobs and his presentation. One more thing, if you have any questions, because Sheena Jocelyn is in Toronto, and it's 3 a.m. right now, so we agreed it makes most sense to just email her any questions, and she's going to respond in this way. Otherwise, after the next talks, if you have any questions about understanding anything in particular, please, we have time for 1 or 2 questions right after the talk. And any other broader questions, please use the app, and we're going to talk about those questions at the end of all presentations. Use the app, and I think it's, what was the sub thing? Activity or audience response under the app, please.
Understanding questions, whatever is.
Yeah, oh yeah, the understanding questions we're going to do without the app, so just raise your hands if you have any. We have time for about 1 or 2 questions after each presentation. Thank you.
All right. Thank you for reminding me that it's 3 o'clock in the morning on the East Coast of America. I'm going to do my best. I'm a little sleepy. But anyway, thank you so much, Luca and Simon, for inviting me. You mentioned if we could do these studies in humans. The work that I'm going to tell you about today in my lab is studying human brain recordings. So single neuron correlates of spatial memory and navigation. Okay, so Sheena's talk didn't really get into the details, but what I'm interested in my lab is to understand the kinds of neurons that we think give rise to things such as engram cells, and to understand in a little bit more, with a little more precise level of precision, higher level of precision, what exactly these neurons are doing as we encode memories and go about our everyday life. In order to do this to understand these sorts of things, you start with animal studies. And if you were to look at the hippocampus, there are 2 prominent electrophysiological signatures of what in the hippocampus in behaving animals. Place cells, these are neurons that represent different locations in an environment, and presumably give rise to encoding not only those locations when rats or humans are navigating, but also when we're encoding memories for those locations as well, and also theta oscillations. So the basis for my research, in my lab we do research in humans exclusively, almost exclusively. We have 1 animal project that we're working on now. But the basis for this work is the work that's been done in animals. And we try to test neurons such as place cells and theta oscillations in humans to go more broadly. So a place cell is a neuron. Each square here is 1 place cell. And each neuron represents, its activity represents, we think, 1 location in the environment with the red, so each square here is 1 overhead map of an environment, with the red region being the area of the environment where that neuron has a place field, because it represents that location when the animals are navigating. We try to test whether such phenomena are present in humans as well. And it turns out they are. But it also turns out that human phenomena such as play cells and theta oscillations are quite a bit different compared to rats. So the focus of the work in my lab is to understand, number one, are these phenomena present in humans? And number two, in humans, do we have more advanced capabilities in some form compared to rats? When I got into this field, I was hoping that we would see something special in the human brain. There must be something in there that's different. And we see features of the human hippocampal system that is more advanced, more general than at least what has been seen before compared in rodents. So that's exciting to me, and that's what I'm going to tell you about today. And why might that be? Well, there's in a nutshell, the human hippocampal system has, throughout the course of evolution, has changed quite a bit compared to rats. I guess I probably don't need to tell this particular audience very much, but throughout the course of evolution, the hippocampus in red in humans got quite a bit bigger, and in particular, the anterior, it rotated. So if you were a rat electrophysiologist, you talk about the dorsal hippocampus and the ventral hippocampus. Well, in humans, as many of you probably know, it's rotated and has more of a posterior and anterior orientation. And that, along with it getting bigger, is a pretty big change. So there's a lot of reasons why you might think that the human hippocampus is distinctive. So I think it's important to study. So how do we do this work? Well, so I'm not a medical doctor, but I hang out with a lot of them. That's a big part of my lab. We work with neurosurgeons and neurologists who treat patients who have very severe epilepsy. If you have a relatively more straightforward case of epilepsy, the doctors can often treat it with drugs or behavioral modifications. But the patients who have very severe epilepsy or complicated cases of epilepsy have electrodes implanted in their brains for about 1 to 3 weeks for seizure mapping. There are multiple kinds of and the reason why that is because the doctors in many cases will resect part of the parts of the brain that are giving rise to their seizures. And if you're going to have part of your brain cut out, you better be really sure that the doctors are cutting out the correct part of the brain, to be blunt about it. So the electrodes are implanted to make more precise maps of the patient's function and of the patient's epilepsy. There are multiple kinds of electrodes that are implanted in these patients, both microwire electrodes that record from individual neurons, as well as these macro electrodes that record something like the electrocorticogram or really a fine-scale EEG sorts of signal. Today I'm going to focus on the data from these microwire electrodes that record individual neurons, and Florian is going to talk about that, and Luca too, later. But there is a lot of author signal that you can get from these macro electrodes as well. And the patients, for the most part, during this procedure, when they're not having seizures, they have a lot of free time. So we have them do different kinds of behavioral tasks on a laptop computer that we bring to them, such as this spatial navigation video game, which I'm going to show you, I think, on the next slide. OK. So we try to make the tasks that the patients do in our lab, I try to make them fun. Navigation is sort of like a video game, and we try to design behavioral tasks in my lab that are a little bit more engaging. One of the tasks that we use that I'm going to tell you about later is this task called Treasure Hunt. It's a spatial memory video game. Those are the two people, Ansh and Jonathan, who helped develop this particular task. Oh, gosh, that's a bit loud. On each trial of the task, the subjects are driving. They're told to drive around an environment, learn where different objects are located, and remember where they're located. And they're doing this with a joystick as if it were a video game. And so they drive around the environment, they go to these different treasure chests, and on this trial the subject learns that the object fan is at this position in the environment here. Then they're given a memory cue. Do you remember where the object is located? And they respond yes, maybe, or no. They indicate with a cursor where they think it's positioned. The reason there's that sound is because this is a beach, and that's the waves behind the people. There's a wave right over here. It gets louder when they're at this end of the maze, of the arena. And then they get feedback based on their response. OK, so they responded at this location and at this location. The shield was over there, which is far away from it, so they got that one wrong. The fan was pretty close to it, so they get 100 points for the fan. And their objective is to get their points as high as possible. And really, this cover story is a way of encouraging the subjects to perform the task as accurately as they can, and to encode where they're located and where each object is located throughout the task as accurately as possible. So what is the brain doing when they're doing this sort of task? Well, for starters, we see neurons, what are called play cells and grid cells. Many of you might be if some of you might be familiar with these kinds of findings, but basically in rodents, a place cell is a neuron that represents one location in an environment, and I showed you those examples of them before. A grid cell, and these cells, these place cells, are often found in the hippocampus. Although there's more and more evidence that they are more broad, which is kind of more broadly found, and I think that's pretty interesting. And then there are these neurons called grid cells, which are found in the entorhinal cortex, and one of the main inputs to the hippocampus as well. So these two findings, place cells and grid cells, are present in humans who are doing tasks like this. And this is consistent with rodents. But we also see all kinds of different, we try to design the analyses in my lab to be a little bit more broad, and we see other kinds of results that go beyond what you've seen before in animal studies. So animals have place cells, grid cells, head direction cells, neurons that represent remote places, and also theta oscillations. And if you fill out this table, we see a number of these findings in humans as well. And some of the human findings are similar to rodents, and others, the ones we're going to focus on today, are more novel in humans. OK, so one of the experiments where we found these human neurons that are a little bit more flexible than what you've been seeing before in animal studies come from this task, where we have subjects navigate an environment, but there are 4 different settings where the subjects can navigate. This is a very simple environment. I guess you would hardly call it navigating. But on each trial, the subject drives down a linear track, and they learned 4 different object locations. So in this one, you saw that the person was driving down, and they learned where a desk was located. Now, during the retrieval phase of the task, they're driven down the environment again, and the desk is gone. And their job is to press a button at the location where the desk had been positioned. So essentially, each trial has the subject driving through it, but it's in the context of the person trying to recall a different memory, a different object location. So there are basically 4 different contexts that the person might be in. And the question, and so what does neuronal activity look like, and does it vary with the different contexts that the retrieval context that a person might be in? So I'm going to show you a series of plots like this. The horizontal axis is the position as the person goes down the track from the beginning to the end. The firing rate of the neuron is on a vertical axis, and you can see this neuron has a higher firing rate at the end of the track. So this is a place cell that activates at the end of the track, and if you look at all the different conditions here, the cell has a consistent firing field that's always located at the end of the track. So this is what you would call a standard place cell, because it has a fixed firing field across the entire environment. But if you look at neurons in the entorhinal cortex, you find these neurons, what we call memory trace cells. And here, for the 4 different object conditions, the firing field of this neuron consistently shifts. So here, what I didn't mention is that the blue line is the location of the object that the person is trying to remember. And here, the blue line shifts between the different conditions. But here, you can see that the firing field of this neuron, which is shown in the shading, in the shaded blue, shifts as well. So these memory trace cells seem to be shifting their firing rate to follow the location of the object that the person is trying to remember, except for object 4. In this case, this neuron does not seem to care about object 4. So this is some sort of the phenomenon in rats. It's called remapping. It changes its firing field as a function of where the person is located in the environment. And this is very different from the classical place cells. And importantly, the remapping that occurs here is not based on some visual cue or anything like that, because the scene looks the same on all the trials as well. And here's another one over here. These cells are preferentially found in the entorhinal cortex and also in the frontal lobe as well. We saw these neurons. There we go. OK. What else does the human hippocampal system do that has not been seen before in rodents? We also find, this is work by Melina Tsitsiklis, who was a postdoc in the lab. And what we wanted to do is to test whether there are neurons that represent not only where the person is located themselves, him or herself, but also where perhaps where upcoming objects are located. As you're driving throughout the environment, something that humans can do that maybe rats are not as good at is, as I'm navigating, if I'm trying to remember the location of a treasure chest that's over there, we thought that maybe the person is focused on the location of the remote treasure chest rather than where themselves are located. So we analyzed the neuronal data not as a function of where the person themselves is located, as indicated with the blue dot, but as of the location of the faraway chest. So in the subjects who do this task, we get a lot of data. We have made it designed it to be a fast-paced task, so we get a lot of different object locations across the environment, And you can see on one trial, there are 4 different object locations here, these upcoming treasure chests that the person is located, is following when they're doing the task. And we binned the subject's firing rate as a function of the locations of these objects, as you can see here. So we made a firing rate map that's based on the location, not where they are actually positioned, but of the upcoming object. OK. This is laggy, so sometimes it jumps around. Oh, it gave the punchline away. OK. So what we did, so for one example, a neuron, this is a neuron in the hippocampus. We can compute firing rate maps. These are both overhead maps of the environment, but you can bin the firing rate for each cell as a function of where the person is located, the subject position, or the position of the upcoming treasure chest. And the upshot is that for many of the neurons in this task, they are not reliably encoding where the person themselves are located, but they instead, in this period of the task, are encoding where the subject's upcoming, where the location of the upcoming object is positioned. So this surprised us at first, because we expected to see a lot of these neurons like this one here that are binned by the subject's own position or classical place cells. But instead, in this task, we saw these neurons that represented the upcoming objects. So this seems to be something different in this task, and it follows where the person is attending to in the future. It's not just a static, it's not a constant representation of their own space, but it seems to be more flexible based on something about attention that the human brain can flexibly manipulate in order to go better everyday life. Here's another such cell here. Again, the neuron represents the location of the upcoming object, not of their own position. And 20% of these neurons in the hippocampus and surrounding regions are representing these upcoming regions as well. How am I on time? Am I running late? 4 minutes? OK. I will go fast. I guess I had enough coffee. I don't know. All right. I mentioned in humans that we have these neurons that I'm sorry. I mentioned in rodents we have these neurons that encode the direction that an animal is facing. In animals, the way that these what are called head direction cells activate in rodents are encoding an allocentric representation of an environment, such as neurons that encode, you might have a neuron that represents the concept of encodes direction like north or south, something like that. Humans also have this ability to represent directions in what's called an egocentric fashion, not based on north, south, east, or west, but if something is in the coordinate framework relative to how I'm facing, where I'm pointing in an environment. So if I'm facing straight and I have a neuron that represents somebody being on my right-hand side, it's like Florian being to my right, that would be an egocentric coordinate, because to the right is defined in terms of my own reference frame. And with Lucas Kunz, a postdoc in the lab, we wanted to test whether these sorts of egocentric representations were present in the human brain as well. Lucas and I designed, this paper was published last year, and we designed a framework for testing whether humans have these neurons that we called anchor cells that encode egocentric sorts of representations. And in order to understand it, in order to test whether anchor cells might exist, we needed to, basically it's based on the idea that if the person is located here, the anchor cell are anchored to anchor cells are anchored to particular locations in the environment. So one such anchor cell might activate when a landmark is to my right-hand side, for example. So Lucas designed a framework to test for these kinds of phenomena, to identify anchor fields, and to plot. And he plotted the here you can plot the tuning of these neurons relative, and the coordinates here are ahead, behind, left, and right. Not north, south, east, west, but they're egocentric coordinates relative to how a person is facing. So this is an example anchor cell that is active when this location here is on my left-hand side. And as you can see from this tuning curve there. So it's a very different kind of direction coding from what's been seen before. And the upshot is that we see many of these anchor cells. And these neurons are often found in the parahippocampal cortex. So this is one example. So this is an overhead map of the arena. The anchor field of this neuron, the landmark of in the environment where the cell activates is over here. And this neuron tends to activate when this location is a little bit ahead and a little bit to the right of where I'm pointing. And that corresponds to a shifting. So these arrows here are showing where that, for different locations in the environment, what is the direction where the cell is most active. So the fact that these lines are curving is what makes these kinds of representations very different from classical head direction cells that have been seen before in rats. Another anchor cell here, has a very different kind of response. This is a neuron that is active when this location here is behind you. So whenever the person is driving away from the center of the environment, this anchor cell tends to activate. And this is very different from a classical head direction cell. So there have been some findings, and I don't have time to get into it here, but these anchor cells are also relevant for memory of these spatial locations as well, and I think that's kind of exciting. OK, the last thing that I would like to tell you about today is a new kind of neural coding that we saw in these neurons as well. So everything I've told you about so far is about the firing rate of these neurons and how that encodes information. We also see that the timing of when many neurons encode information is relevant as well. In order to understand the spike timing, I just want to tell you about theta oscillations. Theta oscillations are these large-scale network oscillations that are present in the hippocampus. More or less, during any any task that involves the hippocampus, the theta oscillation in the hippocampus turns on. They show up at about 8 hertz in rodents. Human theta oscillations are present very reliably when humans engage in memory tasks, hippocampally relevant tasks. But humans have not only these 8 hertz theta oscillations like you see here, but we also have these other theta oscillations at about 3 hertz. And these slower theta oscillations are more prominent in humans than they are in rodents. And it seems to be something that makes the human brain a little bit distinctive compared to rats. So one of the reasons why theta oscillations are relevant is because in rodents, it's been shown to underlie something called phase precession, which is a phenomenon where neurons can encode information in rats about where a rat is located based on the timing of when they spike relative to these theta oscillations. So we designed a framework to test for phase precession in humans, but integrating the idea that human theta oscillations are more sophisticated and show up in this slow variety as well as this faster variety as well. And what we see is that humans do have theta phase precession as well, even though humans have these different theta oscillations that vary in frequency quite a bit. Here are 2 neurons that seem to be showing this phenomenon. So this neuron has the highest firing rate here in this one region here in the southwest part of the environment. And when this firing field is active when the person is going in a counterclockwise direction, as you can see here. And if you plot the firing of this neuron while the person is passing through this place field with a dotted line, then you can have this plot here. The horizontal axis here is the position relative to that firing field. So this is the beginning, and this is the end of the pass through it. And the vertical axis is the phase of the neuron firing relative to the theta oscillations. And what you can see is that when the person is at the beginning of the firing field on the left-hand side, then this neuron activates with later phases, close to 2 pi, if you focus on the middle line here. And as the person traverses further and gets towards the end of the maze, towards the end of the firing field, the neuron activates at earlier and earlier phases, as you can see here. And this red line is a regression fit that shows that phenomenon. And basically, so if you look at the big picture here is that if you look at the timing of when the neuron spikes relative to these theta oscillations, you can get information out of where the person is positioned within this field by looking at the phase, the timing of that spiking relative to the oscillations. So this is a theta phase code for location. And what this indicates is that looking at the firing rate of these neurons is good, but there's additional information to be gleaned from the cell's firing that you can get from the timing, from the phase of the spiking. We also see something that we call phase precession for goal states, but I think I probably should wrap up, and you can ask me about that later, or you can check it out in the paper. But the big picture is that neurons encode other kinds of information besides location based on phase precession as well. So it seems to be a general phenomenon. OK, so I think so that's the talk. I hope I gave you a flavor of these human novel representations that we have. Human neurons not only show all these phenomena that have been seen before in rats, which are really important, but we also show things such as remapping for memory retrieval, these egocentric anchor cells, and human theta oscillations show phase precession, and as well as these fast and slow patterns as well. And before I stop talking, I just want to thank my lab and thank my collaborators and thank my funding agencies. I'm especially really grateful for the people, the clinical collaborators and the patients who work with us. I'm not a medical doctor, but I have the opportunity to work with these wonderful doctors. And it lets us do these studies that I think are important and help complement the rodent work. So I could not do this work without any of them. So I'm grateful to them as well. So thanks. And I think there's a moment for clarification questions before we move to the general discussion, right? OK.
So thanks, Josh. Really great work. So I have a quick question with respect to these cells that code for the location of an object rather than location of the person.
Yeah.
Did they fire only when the person had the intention of moving to these objects, in which case it could be interpreted as predictive coding, or did they also, say, fire for object A even if the person was moving to object B?
For object, you mean are you asking if it's about the identity of the object that is hidden in?
No. Sorry, it is the location, right?
Yeah.
But say that the person was not moving to the location of that object, but moving to a different object at a different location.
There, but I'm going there instead.
Yeah.
Yeah, that's a really good question. We didn't have that control condition in there at all, because we tried to design the task to use the patient's time as efficiently as possible. So in fact, if the person started messing around in the environment and going somewhere else, which might be really interesting, we put little flashing lights, flashing arrows on the screen that says, no, go over there to the chest, because we want to get as many trials per session as we could. So unfortunately, we don't have those data.
One more question, maybe before Florian sets up.
Thank you. Great talk. You talked in the beginning about this difference from the mouse to the human hippocampus, going this posterior to anterior. Can you talk a little bit about the cells that you find, whether they have a particular location in this gradient?
Yeah. So for the most part, in the epilepsy patients that we work with, we tend to get neurons in the anterior part of the human hippocampus, which is analogous to the ventral part of the rat hippocampus. But in the rat studies, if you were to extrapolate from the rat studies, the more spatial cells should be in the other part of the hippocampus that I'm not recording from, posterior in humans or dorsal in rats. So once in a while we get, so basically what I'm trying to say is that in rats there's a strong gradient, right? You get more spatial cells in the back of the dorsal part of the hippocampus than in the part that we record from. We've looked a little bit at that sort of gradient in our work because it really predicts that you should see more place cells at the back of the hippocampus. We have not seen that gradient quite as strongly. So I don't know if it's a real difference or if, but on the other hand, we just don't get as many recordings at the back of the hippocampus because it's smaller, so I can't make that conclusion. It sort of seems like there's a difference, and the gradient is not as strong in humans, but we don't get a lot of sampling there, so I'm not totally sure. I'm not real confident in making that statement. Thanks.
Thank you, Josh, for that very cool and interesting talk. Especially the games are really something else. Yeah, I kind of want to try them out myself now. Yeah. So next time we are going to have Florian, yeah. Any further questions you might have, please use the app for it, and we're going to come back to them at the end of the symposium.
Great. So I'd also like to start by thanking Simon and Luca for having me here. It's great to be in Scotland again. I'd like to talk about a different phenomenon that we observe in these epilepsy patients. So the recording setup is very similar to what Josh just described, so I don't need to get into those details. But there is a phenomenon called, or by now named, concept neurons that we observe in the human MTL. So I'd like to describe that, and then two studies where we try to investigate the meaning or the role of these neurons for memory formation. So next you'll get to, so typically what we do is we have these patients there and we show them pictures we downloaded from the internet, and the next slide will show you a neuron's response to these pictures. You'll actually get to hear the neuron. Every action potential is a click. So here we present various pictures to a patient, and this is one particular neuron. Oh no, we tried this, but now it doesn't seem to want to start. Darn. We just tried it before the session and it was working, but okay, here it is. So every action potential is a click, and you'll notice that this particular neuron has a preference for one of the pictures. So there is some baseline firing, but whenever the picture of Marilyn Monroe comes on the screen, the neuron goes crazy. And this is a behavior of neurons that we find in virtually every patient, and we've tested close to 100 by now in Bonn. This is a graphical depiction of what you just heard. This is called a peristimulus time histogram. So here's the target stimulus. And down here, this would be 6 different presentations. You have the rasters here. Every blue line is an action potential. Down here, we have this stuff summed up as a histogram. And this was a recording back at UCLA where Josh and I shared the patients. Well, in Bonn, we don't have Marilyn Monroe responses, but this is the former director of the clinic, Christian Alger, and we found a response to him. And if you think about what made the Marilyn Monroe neuron fire, then of course there's a large number of possibilities. It was the only black and white picture in the series, but it turns out usually it is actually the identity of the person that we're showing. So in this case, we in a morning experiment, we found a response to Christian Alger, and then we ran a follow-up experiment with more than 100 different stimuli, and the only 3 stimuli that this cell responded to, this is a p-value response score, was his picture, his written name, and his name spoken by a computer voice. So here we have a large semantic invariance. The cell responds to the contents of the picture, the semantic contents. And that's what these cells typically do. This is why we call them concept cells. Here's another example. So this was a case where in the morning we found a response to a picture of Harry Potter, and then in the follow-up experiment, my graduate student accidentally clicked on the wrong picture right next to Harry Potter. This is Ron Weasley, another person from the Harry Potter story. And we can see the cell responds to the written and spoken name of Harry Potter, and then to the picture of Ron Weasley. And it might also have responded to other character of the Harry Potter saga, like, I don't know, Hermione Granger. It might have responded to Lord Voldemort or Albus Dumbledore, or maybe to wizards from other stories, like Gandalf. We don't know that. The reason I'm showing this is that all of these neurons appear to have what I would call a semantic tuning curve, and it can be very narrow or it can be wide. Right? So we have cells that respond to only one particular person and nothing else, and then we have cells that respond to wider semantic categories. Now, why would the brain allow itself these very sparse representations? So we know, first of all, the MTL is what we need for episodic memory. We've known that since HM. We observe that the response latencies, which are around 400 milliseconds, are too late for object recognition. Object recognition has already happened. Conscious perception has also already set in, and these responses follow the conscious perception. If I manage to trick you into seeing something that isn't there, then the neurons will follow that. And finally, we need this sparse code because if you think about what episodic memory is, you simply have to associate certain semantic entities, like who was there, where was the scene, what happened, these types of things. And in order to do that, because you could use a network code, but a network can only do one thing at a time. And in this case, if we need to associate things together, then we need to be able to have neurons fire independently together. Right? And that is why this sparse code is necessary to form these things. So the hypothesis that we're following is that these concept neurons basically provide the semantic content that needs to be associated together in order to form a mnemonic episode. And I will look at 2 different studies in which we try to investigate this further. So this is an associative memory task where we have a 3x3 grid, and we have a number of pictures for which we know we have selectively responding cells. And in this case, there are 3 different pictures in 3 different positions. Back then, when we started this, we didn't have an eye tracker yet, so we had the patients actually confirm the position by touching it on a touch screen. So here we have 3 different pictures. Then there is a distraction task where they count backwards from a random starting number in steps of 3. And then there is a retrieval period where, in random order, they see the stimulus and they have to point to the position where it was. Okay? And we want to compare successful versus unsuccessful memory. So it would be great to have comparable numbers for those two. And in order to do that, we use an adaptive algorithm. So if the patients get too good, we make it harder. And for once, we did that by varying the presentation duration on the screen, but more importantly, we varied the set size, right? So if they got too good with 3 pictures, we would go up to 4, which is already pretty hard. And it turns out that indeed the step size, the set size is what really influenced whether they would remember the stimuli or not. We would always stop this experiment after exactly 35 minutes, and then we would look for selectively responding cells, whether they would behave differently for subsequently remembered versus forgotten stimuli. And here we have 2 neurons from the amygdala and hippocampus with 2 selective responses. But these pictures are pictures of friends and family of the patients, so we had to mask them. So we have these 2 responses, and now here in blue we have subsequently remembered, and in red forgotten trials. And when we sum them up, we see that indeed for the remembered trials there is a higher activity in both cases, right? In addition, we could also look at the different locations. We could see whether we had selective responses not to particular stimuli, but across stimuli to different locations. So here we have both of these units are in parahippocampal cortex. That's where we did find most of them. Here we have a unit responding to this field, subfield. Here we have one responding to these 3 down here. And once again, we could look at the difference between remembered and forgotten. And during the main activation, there was no difference at all. But later on, there was a subtle but turns out significant difference. So now if we look at the population statistics, and we recorded about 2,500 units from 20-some patients, what we see here is for those neurons that selectively responded to a particular stimulus, that indeed in the amygdala, the hippocampus, and entorhinal cortex, there was a significant difference between subsequently remembered versus forgotten stimuli. In other words, the activity of these concept neurons allowed to predict successful memory. This was the case for these 3 regions, but not for the parahippocampal cortex. Now, when we look at these location-specific responses, which were present also in these 3 regions, although not very prominent, there was no significant difference here, but now in the hippocampus we see this significant activation, and it's surprisingly late. In the parahippocampal cortex, because the parahippocampal cortex tends to respond at a much shorter latency, around 280 milliseconds. So this is a rather late effect, but we double-checked it. So, for instance, because this is close to the button press, we also made sure that it wasn't related to that. It's still present when we line it to the button press, right? So it seems to be real. As a control, we used all the other neurons, and you see these are really large numbers, sometimes more than 1,000, right? And all those other neurons do not allow us to predict successful memory. So this really is something that's constrained, restricted to these 2 different groups of specific cells. We have a little activation here, but I would consider that's false positive. And I can tell you later why. Anyway, that was one study, and another study where we looked at this contained a pharmacological manipulation. So we came across a rather old study where Veselis et al. Compared different sedative drugs and anesthetics, and it turns out that propofol, more than all the other drugs investigated, causes what one could call an anterograd amnesia. So if under propofol you learn something, then the recall rate is very low, right here. And it turns out that at our center in Bonn, we use propofol to explain the electrodes for brief anesthesia. So the patients are being administered propofol with or without us, and because of that, it wasn't too hard to convince the IRB that we could start the propofol a little more slowly than usual and run an experiment under low dose of propofol before we crank them up and have the patients go into the OR. So for this particular paradigm, what we do is in the morning, we run a standard screening using between 100 and 150 different stimuli, and then we select our 8 most promising response-eliciting stimuli for which we think we have concept cells, and use 16 control pictures. And once we're done with that, we push the patient bed over to the neurosurgery department, and we enter the room right next to the OR, where typically the anesthesia is started. And at first, we don't do anything but run a mini screening. So we use these 8 candidates plus 8 control stimuli, and we have the patients name these objects whenever they see them. Every object is shown 12 times. This is to see whether the responses are still there and how they are without any pharmacological manipulation. Then we start the propofol. So now they're under light doses of propofol, 1.5 micrograms per milliliter. Patients are usually a little tipsy, they feel well. And now they actually have to run a similar task as shown before. So first they see a stimulus, they have to name it, just like before, and then they see it on a grid, and they have to press the field where it is on the grid and are told to remember that. And unlike before, this is done 12 times, and every item is always in the same location. Under normal circumstances, this would be a piece of cake for the patients. But under propofol, it gets fairly hard. So we simply have them perform this task, then we stop the propofol infusion and wait until the target concentration goes below, I think, 0.5 micrograms per milliliter, which takes about 15 minutes. And then we have 3 different recalls that we ask them. So first we want to know if they remember the very first task, which was without any, without the propofol. So we compare these 16 to those 8 new stimuli that they haven't seen before, and ask them to name that. The second thing is we want to know if they know which of these 16 was actually shown in this associative memory task, to see if they remember the task. This could be solved simply by familiarity. And the third one is to actually ask for the item's location, which could be considered source memory. And it turns out the behavioral data shows us they know very well what they've seen in the first part of the task over here. They're pretty bad at remembering what they've seen in this task, and they're really bad at remembering where the locations were. Okay, so behaviorally this was successful. We were able to cause an anterograd amnesia, which could also be seen as a dissociation of conscious perception and memory. They know exactly what they see, but they will not remember at least a lot of these things. And here are 2 examples. So this is a cell that responded to a movie picture. This is a German movie. So we have a clear response here before the propofol in the mini screening. And now under propofol, you can see that the response is delayed and attenuated a little bit. Here's a different unit that responded to Vladimir Putin. This is a long time ago, but a few years back. Anyway, we have a clear response here, and now under propofol, it's almost gone. It's really attenuated. And now when we look at this at the population average, and obviously this is something that we only do with patients where there is a good relationship, trustful relationship, otherwise I wouldn't want to make them tipsy. So we've done this with 9 patients, and here we have basically the population results. These are averaged instantaneous firing rates across units. And green is the mini screening before the propofol, gray is during the propofol, and the orange stuff is during those 3 recall questions. Okay, and what we see here is across these regions, we hardly had any entorhinal neurons, so I didn't show that. I'm not showing those. But what we see is, in general, the responses under propofol are attenuated and delayed. And this effect is probably least pronounced here in parahippocampal cortex. Sometimes they're completely rescued, like the magnitudes, response magnitudes are typically rescued by the time we ask for the recall questions. The latency, maybe not so much. But anyway, these are the effects at the population average, which are concordant to the behavioral results. But of course, it'll be more interesting to look at an actual correlation across stimuli. And that's what we've done here. So what we show here is the difference in peak activation during propofol relative to before propofol, right? So, and we see that indeed for what's described as item and source memory here refers to these two different questions. So the second one, which was in that task, is what we could call item memory, and then to actually know the position requires source memory. And basically, we just separate the trials in a different manner. And what we see here is that for item memory, we see an attenuation in the peak activation, both in hippocampus and amygdala. And we see it for the source memory only in the hippocampus. The next thing here is the attenuation, because this is the position where the picture is shown. But for the learning task, you don't even have all the information yet, right? Although you might remember from the previous repetitions where it was. But anyway, here we can now look at the situation where all the information is available. We see the stimulus and its location. And in this case, there is no defined peak activation, so we simply took the mean of the first second after picture presentation, and we see that item memory, again, is diminished in hippocampus and amygdala, and source memory only in the hippocampus. And finally, we looked at the response latency. The delay, which again we can only calculate for cases where we have a clear response onset, because if this picture is shown first and then this one is shown, there will not be another strong response. So this is something, excuse me, we could compare. And so a latency effect here was found exclusively in the hippocampus for source memory. Those are the things we observed here. And so to wrap up, we find these amazing concept neurons. We see that their activity for the item neurons allows us to predict successful memory, and for the location neurons in parahippocampal cortex. And since I'm running out of time, propofol reduces these effects in a stimulus-specific manner. And with that, I want to thank my collaborators, especially Zina McKay, who conducted these two studies, and thank you all for your attention.
Thank you, Florian. It was a very cool presentation, especially the pharmacological stuff is quite cool to see. We have time for one question, a brief one, and then we're going to, for the sake of time, we're going to continue right away. Yes, please.
Thank you for the great talk. I'm wondering whether there was some lateralization trends with your various types of cell you presented today. Lateralization between left and right side.
Okay, so in these two experiments, in terms of the memory performance, I know we did look at this for the first study, and there was none. We may or may not have done it for the second one. I would need to double check. I would not expect this to be the case. So in terms of these cells, we find them bilaterally. There's the only time we observed an asymmetry was that neurons in the right but not left amygdala tend to respond to animals as a category response. That is the only time we have ever observed a functional asymmetry in these things. So other than that, both hemispheres seem to be doing the same things. Also, we always have two electrodes in the hippocampus, one in the anterior, one in the posterior hippocampus. And based or in terms of these concept neurons, they appear to be identical. There's nothing no difference that we have ever discerned. But then again, like I said, the width of a semantic tuning curve is not trivial to assess. So it could be that, I don't know, there's more category responses posterior, which could perhaps correspond to larger place fields. That is something we would probably need to look into. But from experience, I would say there is no difference.
Perfect. Okay, so I'm going to talk about hippocampal neurons and how we think they code individual episodic memories. Let's jump right in. There are two possible, so first off, we do know that the hippocampus is of course essential for episodic memory. But we wanted to know is what is happening under the hood, what is the exact coding mechanism that is in play here. So one, if we have an episode such as this, you're in your favorite cafe with your best friend, a waiter's attending a different table, your friend brought her dog, there's music playing in the background, so all these diverse elements. According to a theory proposed by Tyler and DeSena more than 40 years ago, all of these elements are then bound to an index assembly of neurons in the hippocampus. Alternatively, in 2012, Rodrigo Quiroga suggested that concept neurons are tuning every single one of these elements. And in this case, this episode would be coded by the concurrent activity of these specific concept neurons. So in this case, the waiter would be represented by a concept neuron, your best friend, and the music playing in the background. So in the end, we have just a bunch of neurons coding for that episode, but the coding mechanism is vastly different. In one case, we have a conjunctive code representing all of these elements at once, and in the other case, we have a more element-specific code. And we're as well working with, that's not supposed to be in there anymore, epileptic patients. And unfortunately, we can't send them to a cafe every single time we want to test their episodic memories. So we instead have this very simple memory association task. We start off by showing them an animal cue. In this case, it's a tiger. And then 2 seconds later, 2 associate images come on the screen. And the task of the patient is to make up a very vivid story consisting of all 3 images. In this case, you might think of Jennifer Aniston riding a tiger throughout the Taj Mahal. It doesn't have to be plausible, it just has to be memorable. Every single one of these episodes is learned and retrieved only a single time, and the task is also self-paced, so they have about as much time to come up with a story as they want to. And we did this to be as close to episodic memory is in real life as possible. So later during cued recall, we present our patients again with the animal cue, and their task is to retrieve the 2 associate images correctly. We have by now a rather decent data set of about 16 patients, 59 sessions, and 585 neurons that are all located in the hippocampus. And this is the backbone of our analysis. This is what everything is based on. So what you can see here is all an analysis on the level of a single neuron. On the y-axis is the standardized firing rate. And on the x-axis, you can see the different trials, the different episodes. So one of those episodes might be the one with Jennifer Aniston and the Taj Mahal. Color-coded here, you can see in purple, encoding the firing rate, and in red, retrieval. And the way we defined reinstatement is by taking the product of the 2. So a very simple metric. And this might look something like this. And you can see immediately there's not a lot going on in most of the episodes. This neuron is not interested in most of the episodes, but it is interested in one. But intuitively saying, well, there's a lot of reinstatement isn't really cutting it. So what we did was running a permutation test. We took the 99th percentile of that and used that as a threshold. So that looks something like this. And there are 2 important things to consider here. We're not looking at a population level. We are looking at a single neuron. And this single neuron is not interested in memory per se. It's coding for specific memories. And this is an So before that was just an analysis schematic. This is how it looks like in real life. So this is one of these neurons. We termed them episode-specific neurons because we found that they're coding specifically for one episode, sometimes two. This is a raster plot you see right there. On the x-axis is the time, and on the y-axis are the different episodes matched for encoding and retrieval. You can see, again, color-coded the action potentials in purple for encoding and red for retrieval. And right away, you can see in trial 7 and 23, there's a lot of firing of this particular single neuron during encoding and then again later during retrieval. Down here, you see the instantaneous firing rate for all reinstated trials, so for trial 7 and 23. But it looks like there might be an effect or a pattern in there where during retrieval, there's an earlier spike. But this, unfortunately, does not check out, so don't focus on that too much. And here on the right, you can see the reinstatement values. For trial 7, it's a swordfish and a face and a place. And for trial 23, it's a cute duck and 2 places that this neuron seems to be interested in. Another interesting bit of information is, first off, the wave shape is rather nice, so it's an indication that it's indeed a single neuron and not a multi-unit. And the other thing is, this neuron is over the recording of roughly 40 to 45 minutes. This is only spiking about 1,100 times, so it's a very sparse firing neuron. And so far, I've told you these neurons and how we define reinstatement, but we still have 2 important problems. The first one is we wanted to exclude concept neuron activity. So that will be the second coding mechanism. And the other one is, well, we have about 600 neurons. Just by chance, we might find a few of these patterns. So in order to tackle both of these questions, we first excluded all neurons that are firing or they have an increased firing rate at encoding after the cue. This way, we want to chug away, exclude all neurons that are tuned to the animal cue. Right? Because if there's firing and encoding just when they see the cue, that's before the associate images come on the screen, which makes it highly likely that this is a concept neuron tuned to, in this example, for example, the tiger. And we also ran a second-order permutation test just to see how many of these neurons we would expect under the null. And you can see that distribution right here on the right side in blue. And the empirical number of neurons that show this pattern is 141. It's about a fourth of all neurons we can record. So it's a highly significant number that we find. But there are more problems. You might say, well, what about the associate images? We could have this firing pattern as well if we literally have a Jennifer Aniston neuron. Right? Jennifer Aniston is present during encoding and retrieval. The neuron might just care about Jennifer Aniston, and now we have a problem. It's just a concept neuron. So in order to tackle that, we were leveraging the task design. The associate images are either a face and a place, or 2 places, or 2 faces. Now, if we restrict our analysis and focus only on neurons that are responding to, that are firing during a trial with 2 faces, and another trial where they're firing during 2 places, we can exclude concept neuron activity because the stimulus material is simply not overlapping. So we do this, repeat the rest of the analysis just as it was before, and we can replicate our findings. Now, bear in mind that it's a rather restrictive analysis where we need reinstatement at least 2 trials. That's why we have a low number, but this is also reflected in the second-order permutation test, and we find a significant number of those. So that was good, but we wanted to go a little bit more deeper, and we figured this is we assume this is a memory process. So if we focus on trials, on episodes that are later forgotten, we would expect not to find this pattern. And again, analysis is exactly the same, but if we focus on the trials that are later forgotten, we do not find a significant number of them. Actually, if we count for an equal number of hits and misses, we find significantly fewer than we would expect otherwise. So that's all good, but there was one problem. If you want to play devil's advocate, the way we were excluding concept neurons tuned to the animal cue is based on a single trial. So this is not what I uploaded. Okay, we're just going to improvise then. So the problem is right here. And now this isn't working. Oh, this is fantastic. Okay. What if we have a neuron that is just a concept neuron that's focused on the or tuned to the tiger? Now the patient might be looking away or be distracted during the 2 seconds until the associates come on. And now after 2 seconds, they focus on the tiger again. It's a concept neuron tuned to the tiger, and we have a high increase in firing rate. And we would say, well, it's not a concept neuron, this is an episode neuron, but we would be wrong. So because of this, we started a second independent data set where the memory task is pretty much the same. We now have one cue and one associate, but the important new bit is that we have a visual tuning task where we take all the images we've previously shown during the episodic memory task, and we show them repeatedly, 6 times in total. Now, if we have a concept neuron tuned to a tiger or Jennifer Aniston, like Florian was talking about earlier, we would expect after about 300 milliseconds a significant increase in firing rate. So using this task, we can now identify, and if we can identify concept neurons, we can also exclude them from our analysis. And if we do this, the rest of the analysis stays exactly the same, but now we can reliably ignore those concept neurons. And we still replicate in this independent data set our findings of a significant number of neurons that show this pattern. Now it's a little bit fewer than a fourth, as we had before. This might be because this data set is a little bit smaller, so there might be some natural variation there, or we might have missed a few of the concept neurons in the first data set. But however it may, we do find a significant number of those, so that made us quite happy. Another thing we need to tick off is, what if those neurons are not just coding for episodes? Maybe they're coding for reoccurring time points in the experiment. So we were looking for time cells. However, we did not find a significant number of time cells, and the time cells that we did find did not overlap with the episode-specific neurons. The task design isn't optimized to find time cells, so I'm not standing here saying there are no time cells, but the fact that we didn't find a significant number of those goes to show that our neurons that we did find cannot be construed as time cells. And another interesting bit is, if we're looking at the wave shape, so the full width at half maximum of those neurons that show this kind of episodic specific pattern, and compare that with all the other single neurons we have, you plot that as a histogram, you can see in purple, bluish, the episode-specific neurons, and the other single neurons in green. You can see the shift to the right. So on average, they have a wider wave shape, which has been shown to be indicative of excitatory cells. So that's another difference between normal single neurons and these episode-specific neurons, which is interesting, makes sense in a way. So to wrap it all up, we find these single neurons that are coding for specific episodes. They're not coding for memory in general. They're not familiarity or novelty cells, as has been reported before. They're coding for specific episodes. And we find this code on the level of a single neuron, and because we don't take all of the recorded cells into consideration. It's not a population code. And these neurons cannot be construed as time cells or concept cells. And as I said just now, they seem to be excitatory neurons. Importantly, I would like to add as well is, what we did not do here is falsify the idea that concept neurons are coding episodes. What we have found, however, was a memory code that is independent of this concept neuron code, which is episode specific. So I'd like to, the manuscript is now on bioRxiv as a preprint. If you have any questions, and you can find me here, please feel free to ask any questions, or put them in the app right now, or send a message to me on Twitter. And I'd also like to specifically thank Simon Hanselmeier, my supervisor here that helped me organize the symposium, and my other 2 supervisors, Maria Wimber and Howard Bowman. Thank you.
All right. One question over there.
Hi, very nice presentation. I very much appreciate the efforts you've gone through to rule out that these are concept neurons, but I must say I'm not convinced for a number of reasons, actually. Yeah. So one is we know from the work of Rodrigo Quiroga and also Matthias Eisson that once you associate something, the moment I see a stimulus and I think of something related, then just thinking of a concept will activate concept cells. That's something we've shown time and again. And I think it's difficult to control, especially when you ask them to be creative and make up a story, what they might include in that story that could even not be in the picture, right? And that is something that is very difficult to test in terms of the tuning. Right, so it's difficult afterwards to really access the tuning of these neurons in order to identify them as concept neurons. Right, you simply may not have those pictures. May not have had those pictures in there to really show that. And the second point is if you want to make the claim that this is episode specific, I would think that a convincing control analysis would be if you have those three things associated together, just switch one of them and show that there is no longer an activation, that the other two are not sufficient, that it's really specifically depending on all three things combined.
Replace one of the images with a completely different image.
Yeah,
I agree. I agree.
But you haven't done that control.
No, so that control is future work that we were thinking about right now. And the other thing, that's true as well. We have no idea what's happening in the head apart from the neural spiking. We don't know what they're thinking about. So this is a criticism that will always be true. We have no idea what they're thinking about. They might be thinking about Marilyn Monroe, for all that I know, and yeah, that's true.
Yeah, and just a comment. I would definitely not want to make the claim that there is no conjunctive coding in the hippocampus. I believe there is, right? But we were focusing on these super invariant, totally context independent. Actually, I mean, we run the screening in one clinic and then we run the follow-up in a totally different building, totally different surrounding. So they are completely context independent and have an effect on memory, right? I think there is this recent opinion article by Rodrigo Quiroga where he claims that there is no sparse coding at all in the hippocampus, which I think is taking it a bit far, but sorry, no pattern separation in humans. That's his claim. Not at all. I think that's taking it too far, but I think he has a very fair point. If everything was conjunctive coding with the number of separate items that human can name and distinguish, there is a combinatorial explosion that will actually make you run out of neurons, even in the human brain.
Yeah. So I think I agree. You would, if you would have to store all memories you've always had. But we do forget things that just gradually decay over time. Most things that happened yesterday, I have forgotten already. So I think that would be my answer to this. The problem I have, if you only have concept neurons and no conjunctive code to supplement this, is how can I distinguish episodes that are highly similar? And it's true there is interference if you have episodes that are highly similar, but we are still able to distinguish them. And a conjunctive code, even if it's just a temporary one, could solve this. It's not the only solution, but it is one. You can have time cells as well. So yeah, does that answer your question?
Yeah, I think Florian took us right to the general discussion, and the more conceptual questions of what these neurons are actually doing, what they're coding, and so we have a few questions here that were asked via the app. So yeah, please, Josh and Florian, if you want to join me up here. So. We'll be starting with the first question. I think that's for you, Josh, I think, with my half-working brain. For the novel human hippocampal functions, which cortical connections do you think have changed during evolution? I think that's for you, Josh, because you made the link between rodent and human hippocampus. Yeah. But obviously for the others, if you chime in.
Yeah, so in my talk, I talked a bit about human responses being different. And it was nice that that question came up earlier. I had a moment to think about it. So thank you for passing your phone to me earlier. Because that's a hard question. I guess one thing, I think that the hippocampus is, more or less the top of the cortical hierarchy and kind of represents everything, can represent everything that's going on in the brain if you are attending to it. Obviously, the human hippocampus has more visual inputs proportionally than say the rat hippocampus, right? Our visual cortex is bigger. So I think that so visual inputs as well as the frontal lobe, I think, has more inputs in the human hippocampus proportionally, especially given our larger anterior hippocampus and being bigger throughout the course of evolution. And a second thing is not a second difference between the human hippocampus compared to rats is that in rats, it isn't so much about the connections, but what is actually attended to, which connections are most strongly functionally relevant at each moment. In rats, the hippocampus is really focused, shows theta oscillations that are really directly tied to locomotion and moving through space. That's a very strong determinant of what's going on in the rat hippocampus. In humans, it's not all about location only. Theta oscillations related to movement are present, but they're not nearly as present as they are in rats. So I think there's less attention to space. And so humans kind of daydream more, and I think that's daydream is a bad term to use, but we're more flexible and we think about other things, and I think that's what's going on too, to help explain things.
I'd like to add, if I may. So I think another problem in navigation, or another difference during navigation between rodents and humans, is that rodents don't see very well and they're very near to the ground. And I think that is why they have to rely on path integration in order to get their bearings and to return to a point where they started. We know from human experiments, if you blindfold them, they're totally bad at path integration. They almost never get back there. And I think one of the reasons is, so I'm not saying that machinery is not present, but I think it's not really relied upon that much. I think the humans probably do more of landmark navigation. We see something and that represents a concept that we can name. We know whatever, a building is there, and that helps us to get our bearings. So I think that is also a difference that has evolved.
But I also think
history.
You can teach rats to, you can put rats in behavioral settings where they behave, where the neurons will respond like what we see in humans. I talked about those neurons that represent remote locations in humans. If you have a rat confined to one location, but they're watching another rat run around the environment, then the rat neurons will respond to where many of the rat neurons will respond to where the other rat is located, which is pretty analogous to the kinds of remote location cells that we see. So I think that, so I guess my point is that it's not all about the connections, but it's about what you can force. If you force the rat to pay attention to other stuff, then you see closer to human-like responses.
All right. Next question. I think that's probably for you, Luca and Florian. Once you have identified concepts and/or episode cells, can you use the spontaneous firing to infer intrinsically generated memories?
What do you mean by intrinsically generated?
I have no. I didn't ask the question.
So yes, would the question be, if we electrically stimulate these cells, could we induce memories, create memories? So I can answer that for the concept neurons.
Hi, thanks. So I was wondering if you have a time course of the recording, right? And then you may have some spontaneous firing that goes over some sort of threshold where you say, was that concept then spontaneously activated, or is it? Can you kind of identify it from the neural noise, background noise, to say that that was semantic concept that was activated in our brain?
And you mean an internal activation?
Yes.
This time, okay. So
yes.
so like when you have memory consolidation, whether there is a reinstatement or reactivation of these neurons?
Yes.
Okay, yeah, we've investigated that. We have a different study where we create a story around those pictures for which we have concept cells. And then we have the patients learn the story, sleep, and recall the story the next morning. And and then we do spike sorting over the entire night, which is pretty tedious, but we found a way to do it, and it's also available online. So in a nutshell, what we find is during sharp wave ripples, during slow wave sleep, we see reactivations of all neurons in the human MTL, but significantly more so for the concept neurons that were involved in a previous experience. So in our case, we were interested to see if the order was the same as in our story, much like we have a replay of place cell sequences. So because of that, we didn't, or we deliberately went for sealing performance. They always remember the story the next morning. So I can't tell you whether memory would be impaired without this reactivation. But I mean, what I can tell you is this reactivation is more strongly than for arbitrary neurons, for these neurons that were related to a recent experience. And the second thing is the order is not preserved. And that makes sense also in light of the first talk we saw in the session. Typically, the wiring of the cells is pre-existent, right? And so if we have 8 stimuli and 8 concept cells, then they will fire in a predetermined order, and that will not be changed by whatever order we put in our story. That's what we found there. That's a manuscript we've been having under review for a while now, or we've been working on the revisions for a while, but we hope to get it out pretty soon.
Yeah, just something very quickly. So we do have a night recording as well that we didn't get to analyze yet, and it's only for 2 patients. And another thing that I think is related to your questions also, what we mostly focus on in regards to memory is when we have over-threshold firing of these neurons, right? But you can always see these neurons are not silent otherwise. So I think it's a very interesting question, is what is happening otherwise? Is this just not... It's this memory reactivation, but just non-conscious sub-threshold, and I could imagine, but we have no data to answer that question explicitly.
All right. So next question. Sorry, I'm looking at the audience to see whether you know anyone wants to ask a question. Otherwise, over there. All right. So let me read out this question, and then you ask your question. Fantastic. With which Florian, I think that's you again. With which frequency do concept neurons fire during successful retrieval?
I'm not sure what's meant by successful retrieval. So if you show the pictures, right, for instance, in this task that I showed for the retrieval, the image is shown as well. That's no internal reactivation. During retrieval, it's comparable to during encoding, and typical firing rates are, well, they go up to 50 hertz, sometimes from a baseline activity of close to 0 hertz, but the typical case is more noise overlaid. Right there, we probably also, with the monotrodes, the spike sorting is not nearly as good as with tetrodes, polytrodes. So unless we have an extremely clean signal, you can never be 100% sure that you're really looking at a single unit. There will be some false positive action potentials in there. I think that's a general problem for all the human single unit recordings. But yeah, activation rates are around 20 hertz, something like that. If you have internally generated responses, they tend to be a little weaker, but they're also very clear. So this is something that they're super highly significant, and you could just by looking at them, it's right in the face. You basically don't need a statistical test, which means you can use any test and it'll come out 10 to minus 10 or more.
Thank you. I was typing my question, but we were limited in word counts, and I wanted to explain it. I was wondering, what are your thoughts on social place cell, or if you tested for them, and the possibility of, cells that are active when navigating social relationship, and the possibility of them being human-defining, that have been evolved recently through the connection that the hippocampus have with more recently evolved neocortical structures.
I guess it's a question for me.
It's a question for.
Okay. Well, since I started talking, I think that the hippocampal, that the same mechanisms that make place cells can be relevant for encoding any kind of relational information that humans pay attention to. So and I think there's this wonderful example, the so-called stretchy bird paper from Tim Barron's group showed that grid cell representations are relevant for position for spatial locations in this space of stretchy birds of different kinds of shapes of animals, which will make more sense if you read the paper. But the basic idea there is that the same kinds of system for these entorhinal grid cells is relevant for all kinds of spaces. So I think that it's totally plausible that you could represent other information like position in social hierarchies and things like that. So I think it's interesting and fascinating and important to understand how broad these kinds of concepts that we see from place cells are relevant more generally. And I sort of think that there's actually that what you're getting at is a really deep link behind the kind of work that I'm working on and that these guys are working on. That really the same kinds of underlying principles are relevant for space as for concepts. That these aren't two different categories of representations, but it's really one really high-dimensional representation that supports both kinds of information.
Yeah, yes, of course. I'll keep it brief. Yeah, I'd like to second that. There's some really cool work by Robert Mock from Cambridge, who shows that many of these findings that we do get in the hippocampus can be explained by a simple clustering algorithm. So, yeah, I would assume that for different kind of inputs, you'd also get an output that can more elegantly maybe even be explained by a clustering procedure.
All right, I think that's a very nice closing, actually, of the session. Yeah, thanks very much for attending the session. I hope you had a good time. And now I guess it's time for the keynote lecture by Ole Jensen. Thank you very much.